Hi, my name is Megan, and today I'm going to be talking to you about my work looking at the regulation mechanism of the bacterial DnaB helicase in vivo. Steric exclusion and wrapping is a mechanism that engages upon helicase polymerase decoupling to slow down helicase activity. I'm going to be talking about 4 mutants that have been previously identified in vitro to disrupt this interaction. DnaB is known to adopt both constricted and dilated states, and this conformational change has been proposed as a helicase regulation mechanism. We found that all of our mutants stabilized either a partially constricted or fully constricted state, and so we propose that there is a joint mechanism wherein both of these things work together to regulate helicase activity. When we put these mutations in vivo, what we found was that there was a general reduction in growth rate and overall reduced strain viability, suggesting that these mutations are deleterious to cells. When we look at them under a microscope, it becomes clear why. We see both a filamented cell phenotype for most of our strains, which is indicative of overall cellular and genomic stress. This is quantified in C. And we also see a dramatic uptick in the number of DNA breaks and nicks, as indicated by the pink BRDU foci. As you can see in B, we quantified the fraction of cells that contain some type of DNA break, and there's a dramatic increase in the population of damaged cells for our mutants relative to wild type. We wanted to look at the overall chromosome complexity, and so we did a rifampicin runout assay that allowed us to do whole integer chromosome counting. And so as you can see, we see major peaks for 2 and 4 chromosomes for wild type, which is what you would expect for log phase cells. And we see a shift to the right for all of our mutants, indicating both an increase in the total number of chromosomes and asynchronous initiation. Using qPCR, we looked at the Ori-Ter ratio for our mutants relative to wild type. And what we saw was a significant increase in the number of origins relative to the number of termini for all of our mutants except for the R74A. Since our mutants have the same initiation rate, this suggests frequent fork stalling for all of our mutants. We looked at the SOS expression using GFP under an SOS promoter, and what we saw under ideal laboratory conditions was that the double mutant has a significant expression of SOS proteins under ideal conditions. This is mirrored in the mutagenesis assay, where we see that the double mutant has a high chance to develop mutants that are resistant to rifampicin. This is likely due to the DNA damage tolerant proteins produced by the SOS response. However, we also see a high level of mutagenesis for our K180A mutant, which is not reflected in the SOS expression, and therefore must be due to some other type of genomic instability. With that, I'd like to thank you all for coming to my talk. I appreciate any comments or questions. We are interested in looking at a REC-A knockout strain with our mutants, looking at the association of other proteins at the replication fork, and looking at how our mutants respond to targeted DNA damage.